Our guest today is a distinguished professor at the University of California, Davis. He received his Bachelor of Arts in Chemistry from Oklahoma City University and continued his education to receive his master's and PhD from Purdue in biochemistry. Throughout his career, he's received a multitude of honors and awards, including chairman of the Gordon Research Conference and also president of the American Society for Nutrition. His career has been devoted to over 40 years of research, and he's focused mainly on the biochemistry and nutritional regulation of vitamins and minerals, especially on the role of nutrients in early growth and the physiological role of cofactors derived from tyrosine. And this has led to over 230 publications from our guest today. If you wouldn't mind, please join me in a warm welcome for our guest, Dr. Rucker, for his first time ever in College Station, actually, as he talks to us today about the nutritional importance of presumed non-essential biofactors in foods with a focus on mitochondriogenesis.
Well, thank you. It's a delight to be here, particularly with so many friends in the audience. This is my first trip to an A&M, but I know at least 10 people here, either because they spent some time at Davis or we were collaborators. And part of my visit today has to do with collaboration with Dr. Choudhury, who has an interest in part in biofactors and foods and things related to energy regulation. And one of our favorite compounds is pyrroloquinoline. And so when I sent in an abstract, I was going to make it a little broader, but I thought it might be well to use PQQ, pyrroloquinoline, as a case study. It's a compound that came on my scene about 25 years ago. And so what you're going to hear in the next hour, or at least I'll attempt to do it, is to give you a 25-year summary of the journey with this particular compound. It started out as a back burner research project in the lab, and I'll spend some time talking about potential mechanisms, its biological importance, and also emphasize some of the lessons that we've learned along the way as it relates to focusing on a biofactor in foods. My early career was very much like Dr. Harris's. What paid the bills in the lab was work related to extracellular matrix proteins. The first 10, 20 years, it was elastin for me, and collagen cross-linking in chemistry that had to do with matrix stability. And there was an enzyme, lysyl oxidase, which was also very important to ED, copper-containing enzyme, that led actually to this interest in pyrroloquinoline. So in that context, some of the lessons were hard ones. So I will talk about some of the lessons learned from this series of studies. Introduce you to some PQQ basics, because as a biofactor, it's really a very interesting, in the true sense of the word, unique compound. So I'll go over some of our past efforts along with more recent studies on its mechanism of action. And so, as I mentioned, PQQ is unique. In fact, it's been around since our earliest evolution. You can find PQQ in stellar dust. Some work done over the last decade by folks at NASA, particularly as it relates to the spacecraft Stardust project. There's a mass spectrometry associated with this project has identified a number of amino acids that exist in stellar dust. That was known. It turns out many of these amino acids can annulate. In the case of PQQ, an annulation between a glutamate and tyrosine to form this very interesting orthoquinone compound. And in that regard, it is unique. You can find it everywhere. I don't think we've examined a tissue, either animal or plant, where we haven't seen some PQQ. And if we went out and dug up some soil, you would find that it's a reasonably good source of the compound. It's unique in the fact that it has a very high antioxidant potential, and part of this potential comes from the fact that it can carry out continuous redox cycling. This is a reaction that some of you will recognize as a Strecker reaction. At least many food chemists describe this kind of a reduction modification of amino acids as a Strecker reaction, particularly if you have some sort of catalyst that you can leak to an oxidant. In this case, superoxide anion. If you have a way of adding a reducing agent to that, one can measure this type of cycling along the way. And if that reducing agent is nitroblue tetrazolium, you can measure formazan. And so these assays described on this particular graph were done in part with nitroblue tetrazolium. One can also use fluorescent compounds as reductants and increase the sensitivity. So this in effect is can be used as an assay for PQQ. Whoops, sorry about that. Can be used as an assay for PQQ if you do some column chromatography as a cleanup. And the take-home message from this particular slide is that if you compare PQQ in terms of its redox cycling potential, probably best stated, perhaps antioxidant potential because it can redox cycle. Here is ascorbic acid and a number of other enediols on the right-hand side of the slide, and the relative signal might range from 4 to 50, where PQQ is 20,000. And many of the flavonoids that I know some of the folks in this room are interested in fall somewhere in the middle. So it carries out really very efficient redox cycling. Another important point to make about PQQ, which will become obvious in a minute, is that it can carry out oxidative deaminations. And as shown at the top is deamination of anything with this sort of general structure producing an aldehyde. And it can do this spontaneously at rates faster than most enzymes carry out oxidative deamination. Another example from Churchill's work published some time ago, but indicates that PQQ in the presence of pyridoxamine, for example, chemically in a test tube can produce pyridoxal. Again, the take-home message is that conversion is much more efficient than most of the enzymes involved in modifying, for example, pyridoxine to something like pyridoxal. And to sort of link together and some past history, there are a number of papers in literature where one can take precursors to collagen and elastin and actually make elastic films. And if you want to cross-link them, the precursors, instead of using one of these modifying enzymes, one can just add PQQ and some copper and elastin, tropoelastin, for example, a precursor to elastic fibers, will cross-link, and one can make elastic films, which are now used for all kinds of surgical applications. Just to a few more basics, I indicated PQQ is found in soil. It's a fairly potent plant growth factor. It's also made by methylotropic bacteria of various types, and rhizobacteria, common soil bacteria. I won't go through this slide other than just point out that there are synthetic pathways for PQQ in methylotropic bacteria. What I will point out is that many other organisms don't make PQQ. For example, E. coli is unable to produce PQQ as a cofactor. Yet, in E. coli, there are a number of dehydrogenases where PQQ is a redox cofactor. And if you grow E. coli in culture and you provide PQQ, they begin to express these so-called quinoproteins over the NAD requiring dehydrogenases to do things like oxidize glucose. And as a commercial note, one can probably go down to CVS and order a glucose assay kit that has recombinant, I assume it's a recombinant enzyme, glucose dehydrogenase from E. coli. that's PQQ dependent, and it's a very sensitive assay for glucose. One of the many kinds of enzymatic assays, and all you have to do is couple it with a dye, and you can measure actually a large number of compounds. Okay, so that was in part where the an interest in PQQ. That's in part where the work was up until the late 1980s. It was recognized as a new cofactor. It was primarily viewed as a cofactor for vitamins, and there were a couple of papers that were published in the mid 80s that suggested that PQQ was a cofactor for, again, Ed Harris and my favorite enzyme, lysyl oxidase. And these papers were based on absorption and resonance spectra, particularly Raman spectra, that were identical to the Raman spectra for PQQ. Many of the a large class of amine oxidases, at one point in time it was thought that they would be 6 dependent. Then it wasn't clear what the carbonyl cofactor was, and these papers began to suggest that a covalently bound PQQ was the cofactor for lysyl oxidase. And I mentioned lysyl oxidase can carry out the oxidative deamination of terminal lysyl groups, and then a lot of magic occurs where if you have big thumbs, this is hard to function, where a lot of magic occurs wherein non-enzymatically there are these aldol and shift-base type crosslinks that occur, and with further maturation you can even get pyridinium compounds. So this cross-linking scheme is very important to collagen and elastin, and it as soon as I gave some thought to these papers, being in a department where nutrition and development was a major theme, it seemed to me that if PQQ was a cofactor for amine oxidases, this would suggest that it might indeed be a new classical vitamin. And so we did a number of experiments, and being again an environment where nutrition and development was a feature of the work, we designed diets that were relatively simple. They were amino acid based diets. In the initial studies, we actually grew the animals in isolators, and we had that redox cycling assay that gave us crude measures, at least, of what we thought was PQQ at the time. And we decided to look at animals over several generations. This is actually an F1 generation. So we would impregnate the animals, assign them to either a deficient or a supplemented diet, and examine then the offspring. And what we observed was that many of the offspring had very friable skin. They had this kyphotic appearance. That kyphosis in a mouse was oftentimes referred to as a Hurley sign. A colleague at Davis, that's sort of it was a pejorative somewhat directed at one of my colleagues who looked at mice in various types of situations. Anyway, they had skeletal problems. And when we looked at issues like lysyl oxidase, we had an antibody so we could measure both activity and amount of protein. It was decreased. If there's less cross-linking and things like tissues like skin collagen, there was an increase in collagen solubility, and that's a crude indication of degree of cross-linking. And we could measure some of the so-called lysyl-norleucine crosslinks, and they were depressed in these animals. So we, at this point, thought, maybe PQQ is a vitamin, and made that suggestion in an early paper in Science. There were a couple of lessons learned from that. One, I was called by one of the editors in Science that said, you know, get busy. The next month is going to be, you're going to be on the phone a lot, because I've read your paper and it's very interesting and it seems to be very timely, and I can understand it. And if I can understand it, then science writers can understand it. So they'll probably be giving you a call. The paper was published, and about the day that it was published, I also got a call from Judith Klinman, who some of you may know. Judith Klinman is at the University of California, Berkeley, and a really superb biochemist who has done a lot of work on copper-containing amino oxidases, among other types of enzymatic systems. And what she was able to show was that the role of copper in these oxidases was not so much its function was not so much to interact with oxygen in the context of an oxidative deamination, but its real function, one of its real functions was once it was placed in the enzyme appropriately, it acted as a Fenton catalyst to create this compound, trioxyphenylalanine, peptidyl trioxyphenylalanine, referred to as TOPA, and provided some very convincing evidence that this was the cofactor for lysyl oxidase. So here we were, we had a paper in Science, an interesting animal model. All sorts of awful things happened to these poor little mice that we fed this fish and diet to. And PQQ was not the cofactor, nor could we use it to explain why we were seeing this particular phenomena. So that moved PQQ up a bit from a back burner project to one that we began to do on a more serious basis, just to see as a biofactor in foods what its mechanism of action was. And I'm going to go through a few slides here just to indicate some of the protocol. As I mentioned, in the initial studies we used amino acid-based diets. Well, as I'll mention in a moment, we improved on assays so that we could measure PQQ with a little more precision than the redox cycling assay. And we could usually engineer a diet, or if we found some casein, we might substitute it for the amino acid mix that contained literally femtomoles of, or picomolar amounts at least, of pyrroloquinoline quinone. So it was a diet devoid enough so that we could actually carry out the experiments. In the experiments for the Science paper, we had added antibiotics because we weren't quite sure whether or not it was made by intestinal microflora at the time. And we were less rigorous with the use of something like an isolator. So from this point on, the animals were grown in a somewhat more conventionally, although the diets were carefully prepared. And the protocol often was to have virgin dams, either mice or rats, adapt them to the diet, and the diet being devoid of PQQ. In the early days, we would add maybe 2 milligrams per kilogram, not very much actually. And then we would maintain the progeny with the diet fed to the dam, and we examined the F1 progeny. And some of the initial experiments, we tried to look at second generation as well. And what we found was that if you looked at the animals that were the dams, for example, if we looked at F0 generations, there were a few demonstrable effects and more or less normal growth. As we began to examine the model more thoroughly, we later found a number of metabolic defects that we could discuss. But in terms of reproductive performance, some of the data are shown in this small figure. What we found was that in this generation, there were signs of gestational and neonatal deficiency, very much like it showed in one of the previous slides, certainly slower rates of growth. And the pups weren't very viable if they were fed this PQQ deficient diet. And if we took these animals, the first generation, and bred them again, then there were few, if any, surviving young. So the model we felt was an important observation. Was a new set of students were able to replicate it. And This summarizes some of the earlier work. Francine Steinberg, who's the current chair of the department at Nutrition, was at Davis, was a student in my lab, and this is a chapter out of her PhD thesis, at least some of the experiments as a part of that. They were important experiments in that they showed that if you added dietary PQQ in amounts that range from 0 to 1 or 2 milligrams per kilogram of diet, that optimal growth occurred somewhere around 300 micrograms. And we got excited about that. That's close to the requirement of folic acid, for example. It's in the range of compounds that we know are essential for other reasons. There was eventually some catch-up growth, but we carried many of these experiments out to weeks 20 and 30. And if you looked at things that develop and get expressed early in development, for example, issues related to T cell or B cell development, if you would take samples of blood and introduce various types of mitogens, Con A for T cells, or various types of lipopolysaccharides to excite various types of B cells, what we found was that the relationship, even in animals that were repleted at 14 weeks, that this was a persistent memory, and somewhere around 200 to 300 micrograms of PQQ in the diet, you could get this kind of growth response curve that had to do with either B or T cell activation. And we re-examined some issues related to collagen. This is a slide that is the expression of pro-chains, pro-alpha-1 chains of collagen. And one could tell that as it relates to weight of the animal, there was a very good correlation between weight of the animal, and all of these data points are from PQQ sufficient animals. That what we were really looking at in that early science paper was the responses that had more to do with early growth and development than they had to do with, say, some change in lysyl oxidase. And I could show you a lot of data here. This is just another figure that indicates that when we looked at lysyl oxidase, either activity or as protein versus expression of pro-collagen alpha-1 chains, there was a nice correlation. So this was work that was done to help clarify some of the details that we left out of the science paper. A lesson learned in part that one's enthusiasm sometimes needs to be metered a bit as it relates to making sure that you have, for want of a better phrase, all your ducks in a row. And there was another student in the lab, Tracy Stites, who made another observation. I won't go through all of Tracy's data, but one of the things he observed was that if you looked at sort of even light level morphology of liver cells, there appeared to be fewer mitochondria. And some more data are shown in this slide. And what we were able to do was sort of classical mitochondrial characterization. We measured respiratory control rates, for example, keto ratios and the like. And appreciate these are very neonatal animals. And so even though the RCR or the respiratory control ratio, normally you'd like to see around 6, 7, 8, 9. In neonates, mitochondria tend to be a little leaky. So these were the values that we got. And these values are really compromised because we elected not to use preparations where the respiratory control rate was less than 2. So the real take-home message of this slide is if we looked at the percentage of functional mitochondrial preparations, the animals receiving PQQ, about 80% of the preparations were fine, but we could only get 30%, or we only studied out of the preparations we had, 30% of them because they had this RCA-R ratios that were greater than 2. And again, the number of mitochondria per cell, we were able, using a variety of techniques, by this point, we used some mitochondrial DNA markers, and we could, by a variety of techniques, begin to be able to show that the number of mitochondria were indeed reduced. So that provided a certain amount of background. And there were other questions we asked during this period that we felt were important in terms of making some nutritional decisions, one of which is PQQ made in the intestine. And I really don't have time this afternoon to go through that data, but the short answer is no. E. coli don't make it, and we had ways of labeling it, and Karsten Schmidt did a very nice job of looking at synthesis of PQQ both in a variety of bacterial systems, and we could not show that it was made in the intestine. That was important because if we get it, then it means that diet is probably, if not the most important, certainly an important source of PQQ. The next question we asked is whether or not PQQ is easily absorbed, and the answer to that was yes. We did some collaboration, and another take-home message was how important networking is. It's very hard to do these kind of broad-based studies unless you get an awful lot of people involved. And there were some folks at the National Lab who could make C14 labeled pyrroloquinoline quinone from one of the methylotropic bacteria strains. So we had access to C14 pyrroloquinoline quinone, and we're able to show that it is easily absorbed. In fact, if we gave large doses, we could recover much of it in the urine, which suggested the animal was certainly capable of absorbing it. And the other thing we did, and this was in addition a lesson learned, is we refined our assays and moved away from some of the chemical assays we were using. And that certainly was not a mistake, but it narrowed the range of compounds that we looked at, and that'll become important in a moment. The assay that we developed, and it from others who had provided suggestions to literature, was to make a recombinant glucose dehydrogenase. And so then if you had a system where you added some glucose and an appropriate reducing agent where you can measure a signal, it turns out that the amount of PQQ that was, it added to the preparation. The catalyst here was the limiting factor, and so it made for a very nice assay. So when we did tissues, we oftentimes would go through a protocol and use the enzymatic assay. The other thing we developed was a mass spec assay for PQQ, and the lesson learned was that we ignored an important PQQ intermediate. And I know there's 3 or 4 people in this room who, including students, many more, that have an interest in various types of biofactors in foods. And many of these biofactors have dozens of derivatives. And so one of the questions is it the parent compound? Is it a derivative? Is it a modified compound that's active? It turns out if you measure this imidazole derivative of pyrroloquinoline quinone, there's a fair amount of it in tissues, and the dissociation constant is fairly low. So we never really had good estimates of total. And PQQ is so reactive with amino acids and proteins, just going through certain isolation protocols, you can reduce or increase it depending upon factors like pH, for example. But the more important take-home messages from this slide is you can find IPQ in a number of tissues. This is a concentration in human milk. And here's a peak for IPQ. PQQ is this peak here. If you add them together, it turns out, based on amounts per, say, kilogram of milk solid, or you can even express it as kilocalories needed in the context of some sort of animal model. The amounts that you find in human milk as PQQ plus IPQ, and IPQ is probably the compound that's absorbed, particularly if you feed it in a diet because it is so reactive with free amino acids and proteins. that amount, it turns out to be about this number of 300 to 400 micrograms per kilogram of solid food. So there is certainly enough in human milk to meet the requirement for a mouse. And if you look at PQQ in human tissues, here's some values from a group that also used mass spec to look at the amounts present. We've done similar assays for the mouse. Our data correspond very similarly, except they're on the order of 4 to 8 times greater based on that GDH assay. And even greater still if you do a chromatographic isolation and use the redox cycling assay. My point here is that if I had to do this all over again, I would have probably relied more on mass spectrometry so that we could work out some of the subtleties as it relates to perhaps important derivatives and factors related to their metabolism. So with that as a background, we then sort of changed directions, and we didn't change directions. We said as a next step, we really have to get a handle on mechanism. And I've listed a number of papers, I think some of which were probably provided to some of you. Again, networking is important. Each one of these first authors or folks associated with authorships are, it came to my laboratory at actually about a time I was beginning to retire. And so it was all fortuitous as it relates to being a part of one network or another. A situation of which I'll point out, sometimes doing research can result in various sort of fortuitous arrangements. Esqui Chaparian had a master's degree from my laboratory and went to UCSF in San Francisco, and stayed there for a while, and then worked for a company, a biotech company that was eventually, Telerik was the name of the company, that was eventually purchased by Amgen. Why is that important? It turns out that Oski received a PhD in my lab. This is part of her PhD work. And but she was a very clever person, and we had developed some networks. So she did her work not on the Davis campus, but it did it at Amgen. And you needed a master's kind of concentration for a PhD at Davis. So she took these courses at UCSF in pharmacology, where some of our contacts were. And the Telerik reference is important in that she called me up and said, you know, I do have some things I can bring to the table, in addition to a very nice salary that Amgen was paying her, as well as some time off to work on a PhD. She said, we have all these code link microarray chips that are available, 300 or 400. Well, these were 10,000 gene array. These chips had were capable of identifying, in the early 1990s anyway, about 10,000 different genes. And the chips back in those days ran about $1,000. And so not only did I get Oski back in my lab vis-a-vis Amgen, there was about $300,000 worth of reagent that came my way as well. And this allowed us to do some very interesting experiments that did indeed set us on a path. The identification really of some transcriptional networks that were important to deciding what PQQ was doing. When Yu Choudhury and Katie Browley came from Bo Lonerdal's lab, there were reasons why a couple folks in here very know Bo very well, and they had some time in Davis, and so there were 2 or 3 years spent where Wen Yu did some really marvelous cell biology, and Katie did some extensions of that. At the end of this, I'll talk about some contributions of Dr. Choudhury from Texas A&M that were experiments that we would have liked to done, and she did them for us, which was wonderful. And we were able to, before I retired to do one human study that will hopefully provide, again, to use the term, some translation as it relates to some of the pathways that I'm going to show you. So many of these slides are busy, and I'll be going through them quickly, but they're important in that if you can look at 10,000 genes, you can make an awful lot of decisions about how things seem to operate. And furthermore, if you have enough chips so that you can look at individual animals in large numbers, it allows you to do some interesting statistics with large databases. So the protocol that we used was plus and minus again. Then we bred the offspring, and somewhere down the line, usually at 5 or 6 weeks, if they were on a minus diet, we switched them, we repleted them. If they were on a sufficient diet, we then switched them to a deficient diet. So the rationale was, and the hypothesis was, is we would get excited about genes if upon deficiency, the signal was increased, and upon repletion, the signal went down, or vice versa. And since the end was large enough, when we did the usual kind of comparisons that one has to do, this is about 10 genes here. I think we looked at least 40. Try to validate them using various types of PCR approaches. As you can, although there are qualitative differences, the pattern of all of these genes, control, deficient, maybe partially repleted, and then controlled and changed to partially repleted or really repleted in some context. The pattern of these genes were very similar when you compared microarray data to PCR data. So we knew we had very good validated data sets, and the take-home message from many of these genes, and if you just read across, many of them seem to be associated with lipid metabolism. So that put us in the direction, and we had the mitochondria data from previous studies that put us in the direction of looking at mitochondriogenesis in a real way. And so we could measure plasma PQQ. What we found is if you had all sorts of measures of increase in, say, mitochondrial number, whether it's enzyme activities like cytochrome oxidase or succinic acid dehydrogenase, or measures of mitochondrial DNA, there was very nice correlations related to vitamin status. One of the genes that fortunately was in the array was a gene was PGC1 alpha, a co-translator facilitator that's very important to mitochondriogenesis. TFAM is a transcription factor that turns on mitochondrial DNA synthesis. If the activation of this particular co-activator is very important to TFAM expression, we could show that indeed they were correlated, again related to whatever dietary perturbation that we were using at the time, particularly in the context of how much PQQ exposure the animals got. And that allowed us to, and then what we were able to do in addition is that we knew that from the gene array that there were very strong signals that were coming out of the so-called Janus kinase pathway, or one of the MAP kinase pathways. These are pathways that, among many other things, allow the phosphorylation of a factor called Krebs. There's a Krebs site on the PGC1 alpha gene, and so one can begin to develop in a stepwise way, essentially a pathway of how these factors seem to be interacting. And we did a number of validation experiments. I'll just focus on this one here. If we used a sort of a promoter transfection with using the promoter for PGC1 alpha, there is this Krebs binding site on that promoter. Krebs has to be phosphorylated, but in this, but I'll come back to that in a moment. If the promoter was absent in the Cre site, then there was not much signal. Otherwise, we got an increase in PGC1 alpha, at least the products from that the promoter was in front of when PQQ was added. So at this point, we had a pretty good idea that PQQ was probably related to one of the PGC1 pathways. And Wen-Yu was then able to show that phosphorylation was an event that was very important to Krebs. Phosphorylation was transient, but if we added the way these experiments were done, they were done with cells in culture, and what PQQ seemed to do was activate Krebs phosphorylation. If it's phosphorylated, then it leads to the next step, which is activation of PGC1 alpha. And if this coactivator is in an activated state, it interacts with other transcription factors such as nuclear respiratory factor or factors involved in mitochondrial organization. And again, to cut to the chase a bit, anytime we added PQQ, we saw marked stimulation in these factors, along with TFAM, the transcription factor that's important for DNA synthesis in mitochondria. So we felt like we had worked out in part a reasonable aspect of a pathway that seemed to be really robust. I'm not quite sure how we got to know each other initially, but it had to do, I think, with some PQQ funding, and anyway, it led to a lot of really wonderful interaction, largely vis-a-vis emails. But part of this interaction resulted in a paper that was published about 6 months ago from Dr. Choudhury's laboratory, which filled in yet another missing piece. And I appreciate I'm going over this somewhat rapidly, and a lot of these components have multiple parts. But another thing that stimulates at least those components that are in the PGC1 alpha pathway are the serotonins. And these are various proteins that act as deacetylases, among other things. It's a large family of proteins. And they often get activated when there are changes in the energy or substrate levels of cells. In a very pragmatic sense, if one exercises, for example, there's a modification in serotonins. They've been associated with aging, and there are a good number of papers on compounds that also work through presumably PGC1, like resveratrol. And so there was a good reason to look at serotins, and Dr. Choudhury chose to look at serotin 1, which is oftentimes associated with cell growth and organization, and serotin 3, which seems to be more associated with mitochondrial activity. And the take-home message from this top row is that if one either looks at protein via Western blotting or transcription of these genes, there's an increase when PQQ is added. What you need to know, and what I've left out, is that for PGC1 to really be activated, there needs to be a deacetylation of type of protein modification that takes place, and serotins are the enzymes that carry that out. And was there protein deacetylation? Doesn't roll off the tongue very well. The answer is yes. The enzymatic, I probably should have shown the pathway and talked about it a bit, but it said serotins work by fairly complicated mechanisms that are NAD dependent. So if you have nicotinamide phosphotransferase, activity, you can alter the NAD to NADH ratio, and it turns out that PQQ tends to elevate NAD, which is a cofactor for one of these serotonins, to facilitate the deacetylation of PGC1 alpha to make it more active in effect. And so it turns out in a similar set of experiments I just showed you, Mahua's laboratory was able to show that indeed, again, in this case, the human cell line, the PCG1 was activated, along with these other factors that are important to mitochondriogenesis and as it relates to mitochondrial DNA synthesis, among several factors. What Katie Browley, who I mentioned, was able to do, and I won't go through all of the, I'll just show a couple of slides. We were able to work with a company called Lipomics, which is located outside Davis. And so there was really a lot of lipid data in terms of lipid concentration. But if in animals that you give PQQ to or make them devoid of it, if mitochondria are going to be affected, one of the things that you would expect would happen is that there would be some sort of perturbation in lipid metabolism. And it turns out, if you look at serum, for example, triglycerides, there were changes in their composition as well as amount that suggested the animals had less mitochondria. And this essentially went back to normal in repleted animals. If you have depressed ability to really deal with fatty acid oxidation, there should be an increase in ketone bodies. Again, there was. And this represents an interesting set of experiments that was hard to do. But we took many of these animals and we had devices so we could actually measure RQ. And if we looked at energy expenditure based on RQ values, fed versus the fasting state, an interesting aspect is the black dots are the animals that saw some PQQ. These are short-term experiments. The open circles are the PQQ devoid animals. One got the impression that when you examine mitochondria, there was a nice relationship between the amount that these little guys would move around in a cage relative to how much mitochondria they had, at least in liver. So there were a lot of things that fit together in those series of studies. I think I can do it easily. In the next 4 or 5 minutes, what I'd like to finish up with is, does this translate into human subjects? A series of experiments that we started 3 or 4 years ago involved volunteers, very much like you folks, and we gave some dietary instructions, although not very strict ones, and then we gave these folks some PQQ, and they took it essentially for about a week, and I should know that, whether it was a week or 2 weeks. They took it they took it for at least 2 weeks, I'm sorry. So we wanted to make sure that they were well repleted, and the amounts they were given was on the order of 10 milligrams a day. We wanted all the tissue saturated, so if there was any kind of, quotes, pharmacologic response, we might be able to see it. And there were a number of things that were observed. If we looked at inflammatory markers, which you might expect to be changed with the consumption of something that had anti-inflammatory as well as antioxidant activity, C-reactive protein was altered. And these are all normal subjects, and these are relatively normal values. If we looked at IL-6, which activates C-reactive protein, a protein that gets expressed as some kind of inflammation that goes on, there were also changes in this particular cytokine. We chose methyl-methionine proteinase 9. It's another inflammatory marker, but primarily for bone disease type disorders. We didn't expect it to change very much, and it didn't. And there was a correlation between the cytokine, which has been related by others to secretion of the C-reactive protein. If there should be some source of validation and should be some sort of correlation, and there was. And this particular individual had a really bad pneumonia into the study, so he was our high-end inflammatory marker. The other was, again, some benefits of networking, and one of my colleagues on the Davis campus was brought in to shore up our impact and ability to do rather large-scale metabolomics. And she's an NMR specialist and was quick to point out to me that she could measure something like 150 to 200 metabolites in a half a mil of urine. So I indicated the experiments we did, and so a priori we did what you're supposed to do when you experiment. We set hypotheses first, and then we set out to see if we could validate them. And one of the hypotheses was if PQQ affects mitochondriogenesis, would you see some changes in the short term in terms of metabolites that might be excreted in urine? And the argument was that if there was some sort of transient activation or increase in efficiency in mitochondria, if you looked at, say, something like TCA cycle intermediates, there ought to be some collective reduction in them. And the answer to that was pyruvate was lower when PQQ was given, lactic acid was lower. And when you looked at metabolites, at least on the right-hand side of the TCA cycle, fumarate and succinate, these also tended to respond to PQQ exposure, a relatively low PQQ exposure. We followed up with some internal controls. As you're probably aware, there are markers of that are often used, creatinine, for example, a marker for sort of muscle-related nitrogen excretion. A more sensitive one is N-methyl histidine. We looked at the correlations of the excretion of one versus the other. We assumed that if we were really getting good data, this correlation should be a good one, and it was. So there was an internal control. And we looked at a number of amino acids in products that are even a little more sensitive than, say, looking at TCA cycle intermediates. One of those end products that seems to be increased if there is an increase in mitochondrial oxidation is 4-hydroxyphenyl lactate as well as acetate. And the answer was that there was an increase in the metabolism of these, expressed by the fact that there were lower levels that we could measure in, say, an external fluid like urine. And trimethylamine and dimethylamine, methylamine are products that largely are derived from the intestine. And trimethylamine is easily absorbed. In fact, most of it's absorbed because the liver gets converted to trimethylamine N oxide. And this is a slide that I was going to change last night because I've left off a few bars, but I think it's probably clear enough. When PQQ was added, there was a decrease in these products. And what I need to tell you is that the enzyme that carries out this oxidation is a PQQ-dependent enzyme, and rather it's an enzyme that increases in response to PQQ. So there is presumably more activity, and we could see this decrease in both the oxide form and particularly that was the case when we looked at the ratio of these two. That's important in that there's one of the things we wanted to assess was whether or not there were changes that were occurring in the intestine. So we looked at a lot of N-methylated products that we knew were derived from the intestine. And we picked this one because we knew it was further modified in the liver, and it appeared the liver was doing more modification than the actual substrate that was being delivered. So other indications that there was altered mitochondrial metabolism came from these NMR studies. And I probably said enough. I've gone through what turns out to be a couple of decades of, or at least brief pictures of experimentation. The way I want to end is we now in the nutrition business, particularly if we're interested in biofactors and foods, there are hundreds that we can choose from. And where we are as it relates to the politics and research of that it's clearly a situation that needs to be changed and focused. And so what I had really kind of hoped this seminar would bring as it relates to that message is that we don't have a very good way of identifying or classifying various types of components in foods. What we do now is we use terms like functional foods or nutraceuticals, and already you wind up quickly over in some marketing domain. So you have somebody who's trying to market some function that's the research driver, and that really never gets you very far in the context of what you would like to know from a basic perspective from which to build a foundation so that you can make real inferences. When I give talks on cell biology and in the class, I oftentimes use this London map in the back and point out the fact that there's a lot of redundancy in a cell. We evolved it from all kinds of different organisms. And that's important if you're doing a cell biology sort of pathway search. The pathway you're on may not be the most robust pathway, for example. But for many of the compounds, and it certainly seems to be true of many of the flavanols and perhaps some of the polyphenolics, is that there are signal pathways that seem to be important to energy regulation. And what I think will evolve over the next 10, 20 years is we'll begin, as we get more information, particularly precise information on food composition, we'll think of many types of bioflavonoids as fingerprints of foods. We've concentrated a lot on macronutrients, and we know a lot about metabolism. My guess is your liver has a pretty good idea when you eat a carrot versus, if you're in Texas, a piece of red meat. And why is that? I think in part it has to do with the kind of fingerprints that various types of food biofactors bring to that picture. We then correlate them with issues related to lipid changes and other things and connect them with some sort of disease process oftentimes or a function. But an exciting area as it relates to any one of the number of compounds that we currently have an interest in is figuring out all the facets of these signaling pathways. That integration, I think, is going to tell us actually a great deal about metabolic regulation in the context of many of the metabolic diseases that we have an interest in. And so with that, I will again say it's been an absolute pleasure to be here. It's fun to talk to folks that I haven't seen, in one case, almost 50 years. That's not entirely true, but it's close. I think we first met about 50 years ago, right? Yeah, too long is the best answer, along with some wonderful students, as I remember them at Davis. You'll have to tell me if they remain wonderful. Thank you very much.
Time for a few questions.
Yes.
Hi, thank you for presenting your work. I was kind of curious about the wet weight levels of PQQ. Given its greatest abundances in the spleen, do you think that PQQ has its greatest role is regulating immune responses?
Well, the only thing I can speak to, there's not a large body of literature by other folks that I could refer to. What I could speak to is our own personal experiences where we could see the responsiveness in a mouse model as it relates to B-cell, T-cell activation. And it's clear also that it's a fairly potent inflammatory. compound. So I'm certain that if one chose the intestine or something like the spleen as a target, there would be some benefit as it relates to asking whether or not there's something efficacious going on as it relates to PQQ. Yeah.
You talked a lot about take-home messages on each one of your slides. If you had to say one about PQQ, what would be your take-home message?
Well, the take-home message is that the question historically with a lot of these compounds is, are they nutritionally important? Are they nutritionally essential? With PQQ, we really set up the experiments so that we could, I think we clearly have done a pretty good job of showing that they're nutritionally important. And to a certain degree, we've indicated if you buy everything that I've told you, there may be some essentiality involved in many of these factors. If there is essentiality, it has to do with the fact that there are extra signals that really help, and I'm talking about biofactors in general here, that when you consider them as a collection of compounds and foods, they help further orchestrate metabolic regulation. So that would be one of the take-home messages using PQQ as an example. If I had to criticize my own experiments, which I think is part of the thrust of your question, we probably haven't focused enough on intestinal factors. Many of these biofactors, PQQ included, are important bacterial mitogens, or indicated plant growth factors, and they'll influence the metabolism of bacteria. So given all of what we don't know about the intestine and signals that come from the intestine, there are probably things there that would be of interest to focus on. Also, it turns out if you use a diet like we have used for many of the studies, that's really pristine. As Ed knows, and it's probably mentioned in the classroom, you can make all kinds of healthful statements that as it relates to lead and cadmium. But if you provoke, for example, P450 system in most animal models, the animals improve a little bit. You know, there's a certain amount of creative tension that comes from that kind of metabolic provocation. So we've looked at a number of epicatechins and other compounds to see if there's a growth response, and the answer is there isn't. So is PQQ unique? I'm still not certain that I want to say boldly yes, because it might imply that it is essential. But the essentiality, I think, is, you know, given it's a real gray area, and the importance of fine regulation is something that characterizes this decade. And, you know, being in the nutrition business, these are factors which seem to really fine-tune regulation. So I've rambled a bit, but that's the take-home message.
Maybe time for one more question.
I was going to ask a short one.
Okay.
There's
Two.
five. So when you looked at the urine, were there protein derivatives, any of the partially isolated? Do you recall?
They're in the paper.
Okay.
Everything we did was in the Harris paper.
There you go.
Yeah.
Okay, one last question then.
You showed some very beautiful data about the PQQ on the mitochondria morphology and the signaling pathway there. So make me thinking about the brown fat thermogenesis. You know that's because it's a lot of pretty hot field in the last few years, and about the mitochondria biogenesis dynamic. So I was thinking about the mitochondrial function. Would you think that could be also applied to brown, like a tissue, like a mitochondria that's like brown fat or muscle?
Mahua may have something to add. But part of this was kind of lessons learned, and it turns out if you use mouse models where brown fat is really important, there are experiments in the literature that you can find with resveratrol. Actually, we've done the PQQ experiments, at least crudely. Many of the flavonoids, you probably know many of these experiments yourself, and if you take mice and feed them high-fat diets, there's a suppression in brown fat, and there's any number of flavonoids that will shift the body composition of those animals just from adding flavonoids. And my guess is that much of it has to do with activation of mitochondria and increased beta oxidation. The extent to which that's species specific is still not clear to me. And much of some of the funding for this work has come from Mitsubishi Gas and Chemical. They make sort of the world's supply of PQQ, and they made a choice about 10 years ago to sell it as a nutraceutical. I was kind of hoping they'd go down the pharma direction, frankly. And so that's only important. And oftentimes I get calls about, mitochondria in the context of exercise and other things. And it's another variable that's often forgotten, and it's animal model dependent as well, is a very important factor in terms of regulating metabolism in general is heat regulation. You only need so much mitochondria. And as it relates to exercise, probably the most limiting nutrient is oxygen. So whether you have a lot, you're really efficient at it, or not so efficient, it's still a matter of, yes?
PQQ is sold over the counter. So the people who take it, why do they take it? For what?
Well, they take it for a variety of reasons. The primary reason for taking PQQ is an argument like the following. One, it's a ubiquitous compound. It has some of the same properties as resveratrol. To get 500 milligrams of resveratrol, if you do it naturally, you probably have to drink a case of really good California Pinot a day. It turns out that in about 3 or 4 glasses of this is, my compound's better than your compound. It's 3 or 4 glasses of a good red wine will probably give you that half milligram of PQQ. So a lot of folks take it as it relates to weight reduction or increased in efficiency, and much of the research in Japan, so there's been a focus on PQQ, has to do with longevity and mental acuity and the like. And the papers, many of which seem to be well done, folks take it for dealing with just mental acuity issues and sense of well-being. There's a lot of things described to it that are inferential. But, I take PQQ myself, and it turns out that my arthritis never expresses itself very much. I'm sore in the mornings, but I don't have to do other things for it, and otherwise it's almost debilitating. So I like the drug, and I've looked at the safety data, and it seems to work in that context. But like a number of folks in this room, there's an awful lot of caution when you pick out some food factor and then treat it like a drug unless you have all the information at hand. It took us a century to get enough information about aspirin. We're still studying it. So, but to answer your question, it's oftentimes viewed as an ergogen.
Okay, well, let's thank Dr. Rucker. We're taking this class for credit. Please make sure you sign. Also, all graduate students are welcome to attend the luncheon with Dr. Rucker immediately following this event, right now, and it's going to be in Cater Matil.